Everyone, and welcome to our presentation. So I'd like to introduce to you Esme and Preston, and they are going to be talking about their research on transcription factors within specific cancer cell lines. So welcome, Esme and Preston.
All right, good morning, everyone. My name is Preston.
I'm Esme.
And we will be discussing transcription factors today.
So we're all affected by cancer. I'd say it's fair to say that almost everyone in this room has had some sort of experience with it, firsthand or secondhand. That's why the field of oncology, the study of the causes, the treatment, and the prevention of cancer is so important.
Cancer can be simply defined as the uncontrollable growth of any of the cells in our bodies. And the causes of this uncontrollable growth are very similar to any other disease or disorder that affects an organism. In that, generally when a DNA sequence that should code for a specific phenotype or characteristic gets mutated, it results in faulty translation and therefore an irregular protein.
There are many different types of cancer, and we looked at a couple specific types for our research, including a couple different cell lines, including PANC1, which is a pancreatic cell line, K562, which is leukemia, C24B, which is prostate cancer, MM1S, which is another type of leukemia, HTC116, which is colon cancer, and MCF7, which is an endocrine-sensitive breast cancer.
With the completion of the Human Genome Project in April 2003, researchers have been able to sequence the entire genetic material of Homo sapiens with an accuracy of 99.999%, which is better than anything anticipated in the initial planning stages of the project. However, not all of the genes have been studied or analyzed specifically or individually, simply due to the vast expanse of the genome itself. And that's where we come in. One of our goals for studying is to contribute to the knowledge of the human genome. And we'll do this by studying a specific domain known as transcription factors. And it's interesting because the human genome, not all of the genes are actively coding for a specific protein. And that's because transcription factors are macromolecules that interact with specific regions of DNA. And they do this by activating or silencing genes. And this happens because they recruit RNA polymerase that leads to translation and therefore protein synthesis. So it's reasonable to assume that the morphological complexity of organisms can be attributed to transcription factors themselves because they're so responsible for cell differentiation, cell growth, and cell development. But with as much power as an inorganic macromolecule, this can lead to things for the worse like cancer. The specific transcription factors that we will be studying are called zinc finger transcription factors, which are unique because they have a zinc ion in the middle that coordinates two other protein structures known as alpha helixes and beta sheets. And the zinc ion combined with these two other structures are actually what allows the transcription factor to bind to the grooves of the DNA and run up and down them to read the DNA. The ones that we will be studying are called GNF277, ZNF274, and ZB2B24. When planning our project, we wanted to answer the questions of what the functions of the three transcription factors are and what their link is to cancer.
And as a subsection of that, we wanted to answer that question by first finding out what cell lines that these transcription factors were expressed in and where they're located inside the cell body. So we used a variety of different methods in order to complete our research, the first of which is cell passaging. Cells are, when they're grown in a nutrient media, and as they grow, for lack of a better term, they basically eat the nutrients out of the media. Also, cells are very particular about how they like to grow. They don't like to be too close to their partner, but they like to be not too far away either. So in order to keep them healthy when growing, you have to take care of them and make sure that they have fresh media as well as they're not overcrowded. And you do this by cell passaging. So you normally will passage cells when they reach 70% confluency, which means that they cover around 70% of the bottom of the dish they're grown in. And so as you can see up there, there's 10%, so there's not a lot of cells yet. 30, 50, and then you would passage it sometime in between where it was in 50 and 90% confluent. In order to passage the cells, you first wash them to get rid of all of the dead cells, and then you detach them from the bottom of the cell dish using an agent called trypsin. Then the cells are resuspended in more nutrient media, and some of that media is transferred to a new dish where the cells will reattach to the bottom of the dish and start to grow. We used this method of cell passaging in order to grow all of the cells previously mentioned and complete our research.
Once we have our cells, we go on to a process known as Western blotting. And Western blotting is an analytical protocol that separates proteins by their size and their charge. The first step of Western blotting is actually to lyse open the cell, or to break it open and extract the protein. And this protein sample is then loaded into a gel with the addition of SDS sample buffer, which is just a dye that allows us to visualize better. The protein sample then gets separated through the pores of the gel, and whatever protein is larger will move slower, and whatever protein is smaller will move faster. And we can separate this with electricity. To facilitate visualization, we will move the protein samples from the gel to another medium known as a PVDF membrane. And this just makes it easier for us to actually study it. But this time the proteins are actually run horizontally onto the membrane instead of down vertically through the pores of the gel. And then once we have the proteins on the membrane, we can take it to a fluorescent imager to actually visualize.
So up here pictured is our electrophoresis. So that was when we were running the proteins vertically down through the gel to separate them. And this is an example of what the gel looks like after we've run it. And so there's us deeply visualizing ourselves. The next step in this process was immunofluorescence, and using the results of our Western blot, we selected two particular cell lines that we wanted to research, and we basically made a marker of the proteins that, or transcription factors that we were trying to research, and we did this by kind of making a stepladder of antibodies and enzymes. So we would wash them, or incubate them in an antibody. First, the primary antibody, which would attach to the transcription factor itself. Then the secondary antibody, which would attach to the primary antibody. And then lastly, an enzyme, which would glow fluorescent when we looked at it under the fluorescent imager. So with our imaging, we could see this cell nucleus in blue, as you can see up there, because of the microscope slide we used, which is called a DAPI lens, kind of. Then beta-tubulin, we stand for beta-tubulin, which is basically marks the cytosol, or the outside of the cell body, and then it showed up in red, except our beta-tubulin stains were fairly nonspecific, and so we didn't see much. And then the proteins that we stained for turned up in green.
We hypothesized that the transcription factors would lie in the nucleus of the cells because it would only make sense that they would localize in the same place as the substance that they would be directly dealing with.
And we also hypothesized that they would have a rough size of 50 to 55 kilodaltons, and that they would be present in most of the cell lines, but with an emphasis on leukemia, because they are supposed to be linked with leukemia. So over the course of our research, we conducted 4 different Western blots. And Western blots are very tedious procedures, and only one of our Western blots was successful, which is pictured up here. This is a Western blot we ran for the transcription factor ZNF277. And we used 2 different antibodies, antibody A and antibody B, just to test the difference between them. And as you can see, there wasn't much of a difference. So they both worked the same. And when we got our results, we were kind of surprised because, as you can see, the blocks that mark the proteins kind of show up in the 17 to 20 kilodalton range. And we hypothesized that they would be more in the 50 to 55 range. This could be for a variety of reasons. It could have something to do, or it could mean these were available cells that are easy to grow.
When we move on to immunofluorescence, we set up a variety of controls, a positive control and a negative control. Our positive control was staining the subnuclear genetic material known as coilin. And coilin from past literature has been made certain that it localizes in the nucleus. So simply because it promotes the assembly of snRNA. And then our negative control was just our DAPI lens, where we just stain the nucleus with no antibodies for proteins. And also pictured is a disrupted nuclei, which represents one of our challenges when visualizing. Similar to the Western blot results, ZNF277 did not image very clearly in when visualizing for immunofluorescence. Only after excessive focusing were we able to pick up the green light emitted from the proteins, and this might not even be indicative of the proteins localizing the cell itself. It might just be the cell autofluorescing, which basically means that natural organisms emit a green light, which ultimately makes our results indeterminate. But it could also be due to non-specific antibodies again.
So with the immunofluorescence of the cell or transcription factor, ZNF274, we were once again kind of surprised when we got our results because we had mixed results. As we stated before, we hypothesized that these transcription factors would be localized in the nucleus of the cell. And as you can see, this was the case in MCF7 by the small green dots inside the blue. But with the HTC116 cell, the proteins seem to be located outside of the nucleus, as seen by the small green dots in the cell body, not the nucleus. This could be due to a variety of reasons. It could be that there's different expression of this transcription factor in different cell lines. It could be that this transcription factor is a different function, or it could also just be due to our own error, once again, where there was disruption in the HTC116 cell nucleuses. So there's more research needed in order to determine this for sure. And then ZBTB24 was the last transcription factor that we imaged. And it confirmed our hypothesis in both cell lines, as you can see by the protein located inside the nucleus in both cells. So this confirmed our hypothesis.
So what we've discussed so far is the prevalence of cancer and the importance of studying it, as well as transcription factors playing a role in possibly the development of cancer, and the specific transcription factors that we've used,
which were ZNF277, ZNF274, and ZBTB24. We used a variety of different methods in order to conduct our research, including cell culture, so the growing of cells, Western blot, which is separating proteins or the transcription factors by size, and then immunofluorescence, which allowed us to locate them inside the cell body. And then we discussed our results, which were, for the most part, pretty good.
Supportive of our hypothesis.
And so although our Western blots were inconclusive, and ZNF277 remains to be a fairly obscure transcription factor, we did confirm that ZNF274 and ZBTB24 are located in the nucleus of the MCF7 cells. And we also discovered that ZNF274 may possibly be located outside of the nucleus in the cell line HTC116. This could be due to a role in differentiating specific types of cancer. So there's more research needed in some of these areas in order to determine and confirm these results.
Yeah, hopefully future research will be able to obtain more viable results for ZNF277, as well as confirm the localization for ZNF274 and ZBTB24, and generally to take cancer treatment to an elevated level with things such as gene therapy. These are references.
For our images, we're excited. And then acknowledgements, we'd
like to thank our mentors, Zach and Alexis, for reminding us of the significance of the project.
Dr. Seth Fritz, who allowed us to work in his lab. I would like to thank the Brian Davidson Health Science, or we would like to thank the Brian Davidson Health Science, Edward Magnan Foundation, Coots and Clark Western Foundation, and the Bacon Family Foundation for supporting us in order to be here.
We'd also just generally like to thank our families for helping us get out of here and do this research.
And I would like to extend a special thank you to Preston, my lab partner, and Zach, who are great bearing with me because I was a little bit clueless in the beginning of this project. And they really taught me a lot, which was awesome. Any questions?